This chapter is going to focus on our adaptive immune system. Our third line of defense is not something we are born with. Rather, it's acquired after an infection or immunization. Our B and T cells go through a rigorous process to respond to a specific pathogen or antigen. Immunocompetence explains the ability of our immune system cells to recognize and respond to a plethora of substances. Antigens are anything foreign that our immune system cells recognize and respond to. Immunogens are antigens that generate a response from our immune system. Virtually anything can be an antigen, including our own cells. Typically, our self cells don't generate a response from the immune system, nor do we want them to. Our adaptive system, or third line of defense, is specific, meaning the antibodies produced can and will only respond to one antigen. For example, an antibody specific for the chickenpox can only work against it and not the measles virus or any other. Another key fact about our adaptive system is that the lymphocytes generate a memory against antigens. The third line is also very diverse and is only turned on when needed. It also has tolerance, meaning it does not react against self cells in a healthy individual. Let's discuss the 4 stages of development of the cells associated with the adaptive system. The first is the development of B and T cells, along with clonal deletion. The second is the presentation of the antigen to the lymphocytes and clonal selection. Third is the challenging, and finally we see the cell-mediated response from the T cells and antibody production from the B cells. First step is the development of the lymphocytes. Both B and T cells arise from the hematopoietic stem cell. The B cells mature in the bone marrow, while the T cells mature in the thymus. Once maturation is complete, the cells migrate to lymphoid organs like the spleen or lymph node. They do this so that they can encounter an antigen and become activated. The lymphocytes are constantly moving in and out of the lymphoid organs. Here we can see our B cell line on the left. Once it matures in the bone marrow, it travels to secondary lymphoid organs, where it will encounter an antigen in the antigen-dependent response. In the third stage, the B cell is challenged by the antigen and is activated, and begins to differentiate into memory cells and antibodies. On the T cell line, we see maturation in the thymus and then a seeding of secondary lymphoid organs. Then we see the antigen presentation and the activation of the T cell. The T cell then differentiates into various subsets of T cells as well as memory cells. So we know that we have our first and second line of defense to protect us from entry of pathogens, but sometimes those defenses get breached. When that occurs, phagocytes make their way to the injury site, start destroying the pathogen, and induce an inflammatory response. Dendritic cells eat the pathogen, if you will, and process it, then display the antigen on its surface. Essentially what it does is take a photo of it around to the lymphocytes, telling them, hey, if you see this guy, destroy it. We also have gamma delta cells that are activated as well. There are different types of markers on the surface of cells that are used for detection, along with recognition and communication. They are all important in the activation of our immune system. The major histocompatibility complex is a set of genes that code for markers or receptors that we find on our cells. They give rise to glycoproteins on our cells other than our red blood cells. They are vital to our immune system recognizing self from non-self, as well as in organ transplants. Class 1 MHC are found on every cell in our body except for red blood cells. Class 2 MHC genes code for glycoproteins on our antigen-presenting cells, which include macrophages, dendritic cells, and B cells. Class 3 genes are associated with the complement system that we discussed last chapter. On your left, you can see an antigen or an MHC molecule on the cell. On your right is a white blood cell that has both MHC1 and MHC2 present because it's nucleated and is an antigen-presenting cell. There are over 300 CD, or cluster of differentiation molecules, that play a role in our immune system. Common ones are CD3, CD4, and CD8. It's the responsibility of the lymphocytes to identify and snatch up antigens. B cells have specific receptors to bind the antigens, while T cells have receptors that can bind to antigens already processed and displayed on MHC. They have the ability to respond to a vast amount of antigens. Once a lymphocyte encounters an antigen and is challenged by it, it makes copies of itself and transforms into different cell types. The cells that are produced are all genetically identical and will all respond to the same antigen. Throughout the differentiation process, memory cells are also generated. The lymphocytes will differ in their responses. After the T cell is activated, there are different cells that it will differentiate into. Helper T cells are the basis of the immune system. They help to activate both B and T cells. Regulatory T cells help to keep the immune system in check by controlling the proliferation and by releasing anti-inflammatory substances. Cytotoxic T cells are the assassins of the immune system because they kill infected cells as well as invaders. T cells are not responsible for the production of antibodies, rather they release cytokines. After activation, B cells produce plasma cells that release antibodies. These antibodies don't kill, but they mark antigens or pathogens for destruction. The maturation and development of T cells is determined by the thymus. CD3 receptors can be found around the T cell receptor and helps in antigen binding. CD4 co-receptors helps T cells to recognize and bind to MHC2, while CD8 co-receptors can be found on cytotoxic T cells and bind to MHC1. Here we see several things on the surface of the T cell. We see CD3. We can see the antigen binding site. We also can see CD4 along with MHC1. Our B cells develop in bone marrow and circulate in the blood to those secondary lymphoid organs where they can encounter an antigen. On the surface of the B cells, we see some differences compared to our T cell. We can see an immunoglobulin with its antigen binding site. We can also see both MHC1 and MHC2. The reason the B cell has MHC2 and the T cell doesn't is because the B cell is an antigen presenting cell. As soon as the lymphocytes have migrated to the lymphoid organs, they already know what specific antigen they're going to respond to. Our genes code for these receptors that we find on the surface of the lymphocytes, and they code for millions of different types. As a result of alternative splicing, we have different RNA transcripts from the same gene. The protein that arises will have variable amino acid sequences and different shapes. Let's discuss receptors found on the surface of B cells. Immunoglobulins are the antigen receptors for B cells and are also antibodies. The immunoglobulin has a distinct structure. At the end of the forks, we can find the antigen binding sites. The variable region will look different on each type of immunoglobulin. They also contain light chains, heavy chains, and a constant region. T cell receptors somewhat resemble B cells in that they're formed by genes. They also have a variable and constant region and can be found inserted in the membrane. They also have a location to which antigens bind and are never secreted. Clonal selection involves the activation of the lymphocytes by antigens. Even before the antigen enters the body, the lymphocytes know the one specific antigen that it can react against. Once they're challenged and activated, they undergo clonal expansion, where they make identical copies of lymphocytes. Clonal deletion involves the removal of lymphocyte clones that react to self. We don't want immune system cells attacking our healthy cells, because when that occurs, we see autoimmune diseases. Those bad guys that do not attack self, that do attack self, are destroyed. Here we see all of the clones. In this image, we can see two clones that recognize and bind to self. We don't want these guys in the picture any longer, so they're destroyed. The good guys survive and then travel to the lymphoid organs, and upon activation can proliferate and differentiate, as we see here. So what exactly is an antigen? In order for something to be considered an antigen, several things must be considered, including how foreign it is, shape, size, as well as accessibility. Immunogens can be an entire microbe or just a portion of it. They can be cells from other humans, animals, or plants. They can also be complex proteins. There are different categories of antigens. Your proteins and exotoxins include enzymes and exotoxins. Lipoproteins found on cell membranes of bacteria are considered antigens. They also include blood cell markers, nucleoproteins, capsules, and the LPS layer of gram-negative bacteria. A good antigen is one that generates a strong response from our immune system by their chemical makeup, cytokines, and size. Generally, larger antigens generate a stronger response, but that isn't always the case. Some good immunogens would include proteins, foreign cells, plant substances, and microbes and viruses. Poor immunogens that generate a weak response include trisaccharides and polypeptides. Haptens are small molecules who alone cannot generate an immune response, but when linked to a larger carrier molecule, they can. The hapten serves as the epitope, or what the immune system cell binds to. An example of this could be a drug or household chemical like soap or detergent. Some haptens generate a response when they bind to serum and proteins. Again, alone, they don't generate a response. Alloantigens are markers found in some members of the same species but not others. These are important in blood groups and histocompatibility profiles. Any alterations in these lead to blood transfusion reactions or transplant rejection. Superantigens are toxins released by bacteria that generate an extremely strong response from T cells. They can activate T cells at about 100 times greater than any other antigen. A significant amount of cytokines released that can lead to death in toxic shock syndrome. We will further discuss allergens in the next chapter. In order for our lymphocytes to be activated, they must encounter an antigen. Macrophages, B cells, and dendritic cells are all antigen-presenting cells. They present the antigens on MHC receptors. Here we see a phagocytic antigen-presenting cell that engulfed a pathogen. During phagocytosis, it broke down the antigen into pieces, which it displays in the groove on MHC2. In this example, we see the presentation to a helper T cell. The T cell uses this receptor to bind to the antigen in MHC2. We also see co-stimulation occurring where CD4 and CD3 also bind. Interleukins are then released by the antigen-presenting cell to activate the T cell, and from the helper T cell to activate other cells. Typically, T cells are activated first. T cell independent antigens are those that can activate B cells without the help of antigen presenting cells or T cells. This can be LPS from E. coli, a capsule, or molecules from viruses. The T cell response is more complex than the B cells. The antigen binding cells determine the actions of the T cells. The T cells must bind to an MHC to be activated, and their cytokines exert a wide range of effects. Activation of the helper T cell specifically is vital for the activation of other immune system cells. There are several subsets of T cells, but I will only hold you responsible for the helper T cells 1 and 2, the regulatory T cell, and the cytotoxic T cell. Helper T cells all have CD4 on their surface and are important in maintaining the entire immune response. They help to activate macrophages through binding to receptors or through the release of cytokines. They activate other T cells by releasing interleukins and can do the same to B cells. They make up the bulk of T cells. Let's look at how T cells are activated. Here we can see an antigen presenting cell bring in an antigen in MHC2. We see the binding and release of interleukins, which activates the naive or young CD4 cell. It then differentiates into the various subsets of helper T cells, which then exert their effects, whether that be activating B cells, stimulating macrophages, increasing inflammation, or regulating the immune system via regulatory cells. A CD8 cell transforms into different T cells. The process is the same, where an antigen-presenting cell brings in an antigen. However, this time it's on MHC1. Helper T cells release interleukins to assist in the activation of the CD8 cell. It then differentiates into a memory T cell and then a cytotoxic T cell, which kills the infected host cells. These guys are great at killing cells. They can destroy cells that are infected by a virus, cancer cells, as well as cells from other animals, as well as humans. The image on the right shows us cytotoxic T cells attacking a cancer cell. Gamma delta cells have T cell receptors that can recognize a variety of antigens. They recognize PAMPs and respond rather quickly with memory. They can also react to tumor cells and are links between the first and second to the third line of defense. In addition to cytotoxic T cells, we have others that are efficient killers. Natural killer cells aren't picky. They will kill anything from cancer cells to virus-infected cells. Technically, they're not part of the adaptive system, but they respond to interleukins and interferon. Natural killer T cells are a mix of NK cells and T cells. They have receptors for antigens and can directly release cytokines. They're another link between the two branches of our immune system. There are two ways a B cell can be activated. They can be activated by antigen-presenting cells or by directly binding to the antigen, as we see here. The B cell engulfs the microbe and displays the antigen to a helper T cell in MHC2. They both release interleukins, and the T cell releases B cell growth factors to activate the B cell. It is then activated and differentiates into the regulatory B cells, memory cells, and plasma cells, which give rise to antibodies. Antibodies have a Y shape, with antigen-binding fragments, a crystallizable fragment, heavy and light chains, and a region that allows swiveling. If we look at the antibody, we can see the variable region, which contains the antigen binding site. We can see the light and heavy chains, and the Fc region. The hypervariable region is where the antibody binds the epitope, or region of the antigen. It varies from antibody to antibody, and can bind epitopes on the same cell or link them. There are several ways that an antibody functions. By binding to the surface of a microbe, an antibody can stimulate opsonization, which enhances phagocytosis. Remember how we talked about how capsules cause phagocytes to not get a grip on the bacteria? This basically puts handles on the microbe for the phagocyte to latch onto. Antibodies can also neutralize pathogens by preventing them from attaching. They can also cause agglutination, which makes the microbe worthless and can enhance phagocytosis. Through antibodies binding and with the complement system, lysis of the microbe can occur. Antitoxins are antibodies that can neutralize the effect of exotoxins released by bacteria. The Fc fragment binds to receptors on macrophages and other immune system cells. By binding to the receptors, either opsonization occurs or the release of chemicals like histamines. Isotypes are different classes of antibodies. IgA can be found in the blood, mucus secretions, salivary glands, and intestines, just to name a few. It resembles a bridge with two antibodies held together. It can also be found in saliva, tears, colostrum of breast milk, and mucus. It provides protection against digestive, respiratory, and genitourinary pathogens. Here we can see the structures of our different antibodies. Here we see IgG, IgA, IgM, IgD, and IgE. IgG makes up the bulk of our antibodies, can cross the placenta, and is released in a primary response. IgM is the first produced upon first exposure to an antigen. IgD is a receptor on B cells, and IgE is produced in an allergic reaction or helminth infection. A titer determines level of antibodies in serum. Research is showing that exposure to an antigen may lead to the production of memory cells that are chemically related, even if that antigen hasn't been seen. This generates a quicker and more effective response the second or third time that an antigen is seen. It also may explain why in some countries, vaccinating for one disease may prevent other infections as well. Let's look at the primary and secondary immune response to antigens. When our immune system encounters an antigen for the first time, it's called the primary response. There's a lag or latent period where the immune system is determining what actions to take. We then see an increase in IgM first, followed by IgG. And then in the secondary response, it could be the second or 100th time that we see the same antigen. In this response, we don't see a latent period. Rather, we see a rapid production of antibodies with IgG produced first and then IgM. You'll notice that the antibody levels remain high, rather than dropping off like after the primary response. Let's discuss immunity and vaccination. Natural immunity is acquired when we get sick, while artificial immunity arises from vaccinations or immune serum. Active immunity is acquired whenever an antigen activates the lymphocytes. It generates memory so that the next time we see the antigen, we can mount a quick response. This requires time to develop, but lasts a long time. It can be generated by natural or artificial means. Passive immunity is acquired when someone receives prepared antibodies. Since the patient has no encounter to the antigen, no memory is generated, and the protection is immediate but doesn't last long. It can come from natural or artificial means. Natural immunity can be active, like when we get sick, or it can be passive, as in breast milk. Artificial immunity can be active through vaccinations or passive through serum injections or antibodies. The first attempt at vaccination took place in 6th century China. They would dry up smallpox scabs and blow the powder into the nose of vulnerable individuals. In the 10th century, dried pus from smallpox lesions were inoculated into healthy individuals. Unfortunately, people still died. Edward Jenner is the father of vaccines. He noticed that a dairy maid who had contracted cowpox was immune to smallpox. He then decided to take the fluid from the cowpox lesions and inject a healthy boy. He then exposed the child to smallpox, and he was immune to it. Naturally, people feared it, but eventually they got over that fear when they proved successful. Passive immunization first involved horse serum, which contained antitoxins to prevent tetanus as well as treat diphtheria. Human products are now used. Gamma globulins can be used for individuals who have weakened immune systems. The whole purpose of vaccines is to challenge the lymphocytes with antigens and generate a primary response and memory. It is done so that upon subsequent exposures, an immediate response can be mounted. They have drastically reduced the amount of infectious diseases that used to be deadly. Just as germicides have desirable characteristics, so do vaccines. They should protect against natural as well as wild forms of a pathogen and have low level of side effects and toxicity. The vaccine should stimulate all lymphocytes and generate memory. We also should not require a significant amount of doses. They should also be affordable, have a long shelf life, and be easy to administer. There are a few types of vaccines. Whole cell or virus vaccines contain live, attenuated cells or viruses. They may also consist of killed cells or inactivated viruses. Vaccines can also have subunits or small antigen components of bacteria and viruses. Vaccines may also be conjugated or linked with proteins to generate a stronger immune system response. Killed vaccines are whole cell vaccines where heat or chemicals are used to inactivate the microbe, but still enables it to generate a response from the immune system, but can't replicate. The hepatitis A and 3 strains of the flu are examples of killed vaccines. Live attenuated vaccines contain live microbes that has its virulence weakened or eliminated. The microbes can replicate and at the same time elicit a response from the immune system. The MMR vaccine, polio, and rubella are examples. There are advantages to whole cell vaccines. The microbes can replicate, produce infection, but don't cause disease. The effects are long-lasting, require few doses, and generate a response from T cells. The downside is that they require special storage and can become virulent again through mutations. Subunit vaccines use portions of the antigen from the microbe. Toxoid vaccines are another example that has a purified exotoxin that has been denatured. Examples of this vaccine include diphtheria, tetanus, and pertussis. No pathogen is present, but an immune response is still generated. In DNA vaccines, DNA from microbes are inserted into plasmids and then inoculated into a recipient. Humans will take up the plasmid and express the microbe's DNA via the proteins. Those proteins can then be expressed on the host cells on the host cells and generate an immune response. Most vaccines are administered via the subcutaneous, intramuscular, and intradermal routes. There are also a few nasal and oral vaccines, and there's research currently being done using a patch. Some vaccines require adjuvants, which enhance the response of the immune system. They help to keep the antigen at the site of injection, as well as keep it in tissues, so that the immune system cells can encounter them. A lume is the most common adjuvant used. It takes years of trials before a vaccine is administered to the general public. Some side effects seen include reactions at the injection site, fever, and allergies. Serious side effects are rare. There's a common misconception that vaccines are linked to autism. The author of a paper claiming they do had his doctor's license stripped, and the paper was removed from the journal. His study was flawed and fraudulent. Vaccines have no link to autism. In 2011, the Institute of Medicine stated vaccines do not cause autism, and in 2013, they reported the schedule was safe and works. Of course, people still believe in the fraudulent claims, so there are a lot of anti-vaxxers out there. This has led to the increase of cases like measles and mumps, which can be prevented with vaccines. The fewer people that vaccinate, there comes a decrease in herd immunity, which means that the pathogen can spread in the population. The more people that are vaccinated, the less likely a microbe can circulate.